Let's talk about testing. Let's talk about specific tests that pick up. Now, we've all agreed now that fusion is the way. The fusion is predictive. Amplification, point mutations are not. What are the tests that are able to pick these up as a foundation? Is it Archer, Karis? You and I are having conversation before on the panel about these different panels. What's the recommendation here? Is it make sure that whatever company you're using is picking this up? Is it just make sure you're doing next generation sequencing and that should capture it? What's the statement that we have here?
Yeah, it's not just a Chem 20. You don't know what you're getting in your Chem 20. So, you know, what we think of, there are 3 TRK fusions, 1, 2, and 3. They're different levels of expression, maybe different in terms of their biology, but we need to, in theory, catch them all. And so most of the groups are working as quickly as they can to dial up their assay so that it captures all of these things. And you can get it from any one of the major players that are out there, any one of the major clinical partners that we all use. I'm not so sure that's true about the internal groups. So the Mass Generals and the Memorials who do their internal stuff, I'm not sure what those panels have. I'm sure they know, but yeah. I don't know.
I can tell you we're not doing it in-house at Hopkins right now.
At Hopkins, so you're not doing this.
No. We are doing it at Penn. We definitely get all of them at Penn.
But John, like, so when I first heard about the drug, and obviously we have to do it, I checked with we're a large cancer center. We have a partner we work with in terms of the NGS, and we asked them, and we got the answer that we will get what we want and all that. But then I had a lot of patients who have done with the same partner for the last 4 or 5 years, but that wasn't done then. So what do? I go back and...
I think that's the question, is do you go back
and I ask do I use the same sample, or some of those patients, obviously, most of them aren't alive, but some of them who are still on treatment or changing treatments, do I go back and say, hey, you have the sample sent to you before, or they don't do biopsy? What would you do?
Well, I would love to hear what the lung people have to say too, because we have lots of tissue usually. Yeah. But would you go as far as rebiopsying somebody on the what is it, 0.2% chance that this would be positive?
I think this. No, I think it's a great question about, we struggle with this all the time when tissue's insufficient, and this brings into the fold the whole idea of plasma genotyping and liquid biopsies. We've got a lot of data in lung recently coming out showing that it can detect alterations that weren't found in tissue if you do it concurrently with tissue interrogation. So,
but you guys are liquid people, so does that work? Are you all getting all 3 fusions?
You get just
off
one.
Of
you get NTRK 1 off a Guardant.
Off a Guardant, okay.
And so
we encourage people to rebiopsy at progression, and I think that's a good thing to do. You know, when you have basket trials out there like NCI Match or ASCO's TAPER study, it allows you to use archived tissue, but I like getting the fresh tissue. And the thing we fall back on is that, you know, Mass General a few years ago reported, you know, the transformation to small cell that occurs 15 to 18% of the time, and that's an EGFR mutant patient. So again, you're looking for T790M, and all of a sudden, wow, it's small cell, totally different therapy you're going here. So we, in the lung field, I think we are trying to retrain. So I don't think, John, to answer your question, we would specifically go after a 0.2% marker, but there are other things associated that allow us to justify getting
And I think it's easier to retest. So in the sarcoma community, it's commonly we retest and we rebiopsy. I think the reason we do it, we also think that the moleculars will change based on prior treatments. I do think it would be a challenge to retest someone who hasn't progressed on a treatment, is doing well, or someone who really, there has to be something that you would think is a good reason. So I think treatment failures is, for us, usually the marker that sort of gets us to rebiopsy and resend.
And I'd just add, I think that Ed also brought in a good point, which is we're not dealing with TRK fusions in a vacuum. And there's so many other, you know, we have the pleomorphic or the heterogeneity in the tumors, and we have all of these other points. So the point is that if we're really doing a good job picking up other mutations, we can do them often together. Yeah. And that brings me just to that point about one of the other tests that we should talk about is there is a development of an immunohistochemistry test. The problem I have with that is I don't think working in sort of like in a vacuum, just looking for TRK fusions is really going to be a viable approach. And the reason for that is the community doc doesn't even know what to do. They're not going to memorize necessarily. I mean, they know what to do in so many ways, but they have to become experienced and experts in so many things now. I think that it's much easier. Let's send a test off, but let's just make sure that test gets everything. And do one test and get everything, as opposed to these like, oh, I'm going to remember on my 0.1% that, oh, I better send off an immunochemistry for this. And then if it's positive, I just don't think that's really, maybe if you had something that you knew was like a 20 or 30%, you wanted to do it as a screening, but I just don't think that's going to happen.
Ben, Ed brought up this point earlier about the culture around rebiopsying and how that was not the right thing to do then. And we've also been through, I think, a decade of when NextGen's been kind of available, or broad sequencing has been available but not paid for. And so there's been this cultural, financial barrier to this. But what we've even seen that kind of go away, thanks to these kinds of medicines that rely on these rare subtypes. Well, it now is part of our culture, just like we get CAT scans on everybody, even though the yield is low. I think we're going to see a culture where we're doing this in everybody. So I'm hopeful that that barrier falling then makes this sort of retesting concept more and more common.
I agree. I can remember NGS coming into the fold into clinical practice probably around 5-7 years ago, and a lot of talk about cost and it being prohibitive. And certainly I think that there's been a greater depth and breadth of what we can find, and it's gotten cheaper. And I think hopefully, again, that will be support for broad use of next generation sequencing.
I mean, the real
Future
question is,
going to be not the Chem 20. Or the blood test is going to be a CBC and the NGS.
Right.
You're right. I mean, that's going to to be part of the base staging, I think.
Yeah, the basic staging
that
of
we're
every
going to see
tumor.
for almost every cancer we
so
take
what
in.
about the provocative question, NGS reflex test and stage 4 disease?
So
What do you think of that? I mean, in my practice, I have to admit I do NGS on stage 4 disease. I do that. I don't do it on stage 3. I don't do it on stage 2 or 1. But I do on every patient who I have. I do NGS on every patient when I first see them.
But I could push back on PANC, for example. We need to know BRCA in everybody. So all of a sudden, we've got some need to do some additional
The other
testing.
thing is that NTRK mutations are driver mutations. And driver genes compete with each other. If one is present, the other one is not present. If we take, for example, colon cancer, maybe in the half of the patients who don't have the RAS mutation, maybe that's more relevant. So we have to think of it also that way, one enrichment. No one has looked into that, but that's a possibility. But again, I do NGS on every stage 4 patient, but the question is it something which we have to talk about like as a more practical thing?